so glad that you're joining us today for the Rena Schechter Memorial Lecture in Cancer Research. We've got an exciting speaker about whom you'll hear more in a moment. Just as a reminder to check out any prior talks you've missed, please head over to the Department of Medicine website, check out our YouTube channel. And as always, for those of us that are on the webinar, please go ahead and send questions via the Q&A function or the chat. I will help moderate those with our speaker later on as we go. Here to present our speaker is Dr. DiPerzio. Dr. DiPerzio serves as the current chief in the Division of Oncology and is the Virginia E. and Sam J. Goldman Professor in Medicine. He's a prolific researcher operating as the director for the Center for Gene and Cellular Immunotherapy here at WashU and is the deputy director of the Siteman Cancer Center. Dr. DiPerzio, thank you for being here.
All right. Thank you, John, for that kind introduction. And I'd like to welcome everyone to the 2021 Rena Schechter Memorial Lecture. And this lectureship was named after Rena Schechter, who was the wife of Sam Schechter, a beloved graduate of Washington University School of Medicine and a long-time internal medicine attending here. Rena was quite a person, and Sam contributed funds for this research fund, this research effort in her name, specifically because of her zest for life, her interest in others, her just unlimiting enthusiasm for patient care. She managed his medical practice for many years. Unfortunately, she died after a long battle with pancreatic cancer, and this lectureship was named in her honor. So this year, we've had many outstanding speakers from around the world come for this lectureship. I mean, looking at the list, I have to say, it's really sort of the best of the best in my view. So this year is no exception. We have Matt Porteus from Stanford University, who's currently, Matt is a professor in the Department of Pediatrics and Institute of Stem Cell Biology and Regenerative Medicine and Maternal Child Health Research Institute at Stanford. He's also co-director of the Stanford Center for Definitive and Curative Medicine. Matt received his undergraduate degree from Harvard, his MD and PhD degrees from Stanford, where his PhD was in the Department of Neurosciences. He went on to do a pediatric hematology oncology fellowship at Boston Children's Hospital and Dana-Farber Cancer Institute, and then a postdoctoral fellowship in the laboratory of David Baltimore, then briefly David Pellman, and then followed David Baltimore to Caltech, where he was a research scholar there for several years. He was recruited to the University of Texas Southwestern, one of my alma maters, and was there as an assistant professor before moving to Stanford, where he's risen to in the ranks as full professor. For anybody who knows Matt's work, obviously he's received many awards. I won't bore you with them. He's very entwined and interdigitated in many of the NIH committees on recombinant molecules. He's been a member of the RAC for many years. He has been one of the leaders in our efforts to move forward with gene manipulation, specifically gene editing. And this was, for many years, gene therapy was something that was actually ignored and poo-pooed in study sections at the NIH, but times have changed. And now we know that we can cure some diseases and hopefully many more in the future using a combination of gene modification, gene editing, base editing, and a number of exciting new techniques which are going to change the face for many patients who have been suffering with these inherited diseases. So without further ado, Matt, thanks for coming.
Thank you. All right. Well, thank you for that kind introduction, and thank you for the invitation to be here. It's exciting to visit WashU for the first time, in particular, exciting to be invited for the Schechter lectureships, and I'm honored to be the next in line of a long list of speakers. Let's see, make sure I got all my tools working. Great. So as John described, the field of gene therapy has exploded over the last decade, which has given opportunities for people like myself to become now also involved at the intersection between academic and biotech research. And I list some of my involvements here. The most relevant of which is Graphite Bio. Now, I'll point out for CME purposes, none of these companies have a commercially approved drug, and I will not be discussing any off-label use of any drugs. I was a history and science major at Harvard, and so I have that sort of as a background. And what I think we all recognize is that with increased understanding of different technologies, we can improve the lives of patients all around the world. Starting with the recognition that sanitation and clean water was a way of improving the health of people, moving in the 19th century, the discovery of antisepsis and anesthesia, facilitating the use of surgical cures for patients. Obviously, the discovery of vaccines has transformed the lives of people all around the world, and in my specialty, of course, in pediatrics, and the last 19 months have reminded us of the importance of vaccines. But I think most of us consider small molecules and in the last 30 years, biologics to be the bread and butter of how we treat patients. And these are tremendously powerful. And of course, the number of lives that have been impacted over history is enormous. But I think we're also, while hematopoietic stem cell transplant has been around for 50 years now, and is a living drug, I think the reality is in the last few years and in the next 20 or 30 years, we're going to see an explosion of cell and gene medicines as a way of treating patients. And I first heard these drugs called living drugs by Carl June, and I think it's a really nice description because these cellular therapies and gene therapies, because they have entirely different pharmacokinetics and pharmacodynamics, that the cells themselves will migrate, respond, divide. They're just very different than a inert substance like a small molecule or a biologic. And I think in the coming years, we're going to also see microbiome manipulation as a living drug as well, although that's taking a little longer to get off the ground. And an example of what I mean by a living drug is demonstrated here, in which we have labeled human hematopoietic stem and progenitor cells with a luciferase gene, and then we injected these cells into the femur of an immunodeficient mouse. And what you can see is at the day of injection, these cells were scattered throughout the body. And then 3 days post, they started to localize, as shown by bioluminescent imaging, to the site of the injection. But instead of degrading now over time, these cells now expand and migrate to the different bones of the body, where they take up residence and start making blood cells. This actually underestimates the impact that these cells are having, because the progeny of these hematopoietic stem and progenitor cells, and specifically the macrophages, are migrating into the tissues, including the brain, where they're having biologic effects. So what we have here is a drug. It took me a while to understand that this cellular product now is a drug that has been genetically engineered and has a distribution that is entirely different than many of our prior modalities. So I am a pediatric hematologist oncologist, and I got into this field because of a desire to improve our treatment for sickle cell disease. Sickle cell disease represents a class of genetic diseases that permeate all of medicine. They particularly permeate all of pediatrics. And when I attended on the pediatric hematology service, the entire service actually could be patients with either sickle cell disease or thalassemia or hemophilia. Really, it could be 100% of the service for patients with these diseases. Most of these, actually all of these diseases in the US would be considered rare diseases. Sickle cell disease is thought to only affect 100,000 people in the US, and we define a rare disease. I think it is, it's got a green light. I'm also happy to use anyway, let me know, I can also use that light. The definition of a rare disease is a disease that affects less than 300,000 people in the US. But in some, these diseases affect millions of people in the US and hundreds of millions of people around the world. Moreover, a single patient with this disease, there's what I call echoes of the disease, because they impact on the parents, the immediate family, and even the community surrounding that patient. And moreover, these diseases affect children. And so one can imagine, if I now can hear that it's on, you can imagine the number of life years saved in terms of decades. And then last year, the group published from NCAT published the National Economic Burden of Rare Disease Study. And what they calculated is between the direct and indirect costs of these rare diseases, not all of them being genetic, but most of them being genetic, was costing the US economy $1 trillion a year. And they estimated that this cost of these rare diseases was equivalent to what cardiovascular disease costs. I'm not sure that's going to be entirely true, but the point is that these, while each individual disease is rare, the impact is tremendous throughout. So in the 1970s, with the development of recombinant DNA technology, it was recognized that one could perhaps use recombinant DNA technology as a treatment for these genetic diseases. And Ted Friedman wrote a short article in Science in 1972, sort of acknowledges maybe the first description of what gene therapy might look like. And then my postdoctoral mentor in the late 1970s sort of described what many people were thinking about, which is that the idea is that there are diseases that affect the blood and brain and other tissues. And he wrote that it'd be a triumph of medicine if we could counter the effects of such genes. And he described for a blood disease that one might take out the stem cells from a patient, modify them in some way, and then give the precursor cells back to the patient and cure them of the disease. So something relatively simple to describe. And yet here we are now 40 years later, and we're just starting to see successes on this vision. So let me go now, spend the next few minutes talking about sickle cell disease, perhaps a review for many of you. So it's I would consider it the most common serious genetic disease in the world. It is caused by a single nucleotide change in the beta globin gene, and adenine is changed to a thymidine. It causes a single amino acid chain in the beta globin protein. The charged glutamic acid, glutamic acid is converted to a polar valine molecule. In the deoxygenated state, this valine molecule contributes to a short hydrophobic surface that allows the hemoglobin tetramers to polymerize. And it converts these pliable biconcave shape red blood cells to the sickle cell red blood cell shape, that gives the disease its name. And these stiff sickle shaped cells block the ability of blood to get to the distal tissues, and so lead to a multisystemic destructive disease that manifests itself in the first decade of life in Africa, where on average most patients die. In the mid-40s is the median lifespan for patients in the US. And unfortunately, this mid-40s median lifespan has not changed over the last 20 or 30 years. So it was first described in the Western world by James Herrick, who took a picture of the blood smear of a dental student of his. But of course, it's been long known in Africa before that. And George Buchanan, a mentor of mine at UT Southwestern, would describe how these African names for the disease have high-pitched vowel sounds denoting pain. And when these names are translated into English, they translate into names like beaten up disease or body biting disease or body chewing disease, I think giving a really vivid description of the destructive property of sickle cell disease. And of course, a number of seminal discoveries in medicine were made by studying sickle cell disease, including in 1949, Linus Pauling describing it as the first molecular disease, Vernon Ingram then identifying the protein mechanism by identifying that amino acid change I just described, the cellular mechanism of polymerization being described in Murayama in 1966, and then the actual DNA and genetic basis of the disease being described in 1977 by a number of investigators at MIT and Yale. And yet for most patients, even up today, the treatments are rudimentary. And I show here a really nice painting by Hertz-Nazaire showing the pain crises and depicting the pain crises these patients have some patients having pain almost every day of their lives, but not coming to medical attention. Other patients having pain, coming to medical attention multiple times a year. And yet, fundamentally, we still treat these patients by time, hydration, and with opiate pain control, with the complexities with using opiates in 2020, the 21st century. We can use hydroxyurea for the prevention of these pain crises, but this is a chemotherapy drug that was discovered decades ago. And we're fortunate in the last couple of years that a couple new drugs have come on the market to decrease the frequency of pain crises. But still, considering the impact that sickle cell disease has had on our understanding of the genetic basis of disease, I feel like our ability to act on that is lagging far behind. So how can we change that story? So the disease, as I said, is manifests from dysfunctional red blood cells. Now, red blood cells don't have a nucleus and have a limited lifespan. So if one were to try to cure this disease for the long term, one would have to focus on the hematopoietic stem cell that gives rise to the red blood cells for the entirety of a patient's life. Of course, the hematopoietic stem cell gives rise to all the blood lineages for the entirety of the person's life. So conceptually, the idea then is we could take hematopoietic stem cells from someone else who doesn't have sickle cell disease and do a transplantation, a stem cell transplantation to a patient who does have sickle cell disease and hopefully cure that disease. And the first example of this was in 1984, where a patient, a 6-year-old girl who was being treated at St. Jude's Children's Hospital, who developed AML. So the indication for her bone marrow transplant was her leukemia. But she had a brother who was an immunologic match with sickle cell trait. And so they did the transplant from her brother and showed that not only did they cure her AML, but they converted her sickle cell disease with high levels of hemoglobin S before transplant to levels of hemoglobin S that matched the levels of hemoglobin S from her brother. And when I was talking to people in the Bay Area a couple years ago about this, they much more in touch than I am and told me that she's still alive and well, cured both of her AML and her sickle cell disease. Now, let's see, 37 years later. And so this first transplant became a basis now for thousands of transplants around the world, allogeneic transplants to cure sickle cell disease. And in 2016, a large group of investigators reported on the overall results. And if you have a matched sibling donor who's an immunologic match and doesn't have sickle cell disease, the outcomes are excellent with a disease-free survival rate in the Western world of 95% or more. The challenge is that only a small fraction of patients actually have a matched sibling donor, that the results with alternative donors, despite extensive research and ongoing research, still have results that are not satisfactory. In any allogeneic transplant, you have the risk of graft versus host disease and post-transplant poor immune reconstitution, which can cause problems. One challenge in the use of bone marrow transplants is these studies and others have shown that the younger you treat the patient, the better the results. But the younger the patient is, often it's hard to, you're exchanging a risk of short-term toxicity with long-term devastation. And it's challenging for patients and physicians and their families to decide when to pull the trigger on this type of therapy. Now, one of the pieces of data that came out of the allogeneic stem cell transplant world that is highly relevant to what I'm going to talk about is that Robert Ione and in a study along with Mark Walters had a single patient who developed mixed chimerism in which the donor cells only occupied between 2 and 5% of the bone marrow. And yet this patient had, for a transient period of time, 50% hemoglobin A and 50% hemoglobin S, and seemed not to have any of the manifestations of sickle cell disease. Courtney Fitzhugh and John Tisdale have shown that perhaps we really need to hit 20% of the 20% donor cells in a mixed chimeric setting to actually cure sickle cell disease. The reason this is important is it gives us a target to shoot for about how many cells might we need to correct if we wanted to correct somebody's own cells and give them back to cure this disease rather than using somebody else's cells, and thereby avoid the challenges of finding an immunologically matched donor and the immunologic challenges of graft versus host disease and immune suppression. So the idea of correcting the sickle cell mutation is again, not a new idea. And in 1985, Oliver Smithies, who won the Nobel Prize for developing homologous recombination in murine embryonic stem cells, demonstrated that he could target the beta globin gene with a piece of DNA using homologous recombination at a frequency of 1 in a million cells. And so there's a couple things about this. First of all, 1 in a million, the finding was important enough to be published in Nature. And I'm showing my age by demonstrating that I actually Xeroxed the paper, which is shown by this little clouding in the upper right-hand corner. I put my name on it because I didn't want somebody to steal my Xerox copy of the paper because I put in a lot of work to do that. And more importantly, there's a little check mark there. It actually meant I didn't only Xerox it, I actually read it, which we don't always do. So this was, you know, the start, but of course, 1 in a million is not nearly sufficient to think about curing any genetic disease. And skipping through a lot of really important developments, we now in 2021 land on this system of genome editing that works as follows, which is we design a protein that has nuclease activity, the ability to cut DNA, and we design it such that it binds to only a specific site in the genome where it makes that double-stranded break. At that point, the cell, the break triggers the cell to activate its repair machinery and repair that break. And there's actually multiple ways that cells can repair double-stranded breaks, but the two major ways are by non-homologous end joining, in which the two ends are stitched back together. Now, this non-homologous end joining process is actually relatively accurate, but occasionally you can get small insertions and deletions at the site of the break. And this now allows both researchers and people interested in developing genetic therapies to make site-specific mutations in specific genes or regulatory elements. But for us, we felt like the thing to focus on was not to make new mutations in the genome, but instead to fix mutations. And for that, we tap into the other way that cells repair double-stranded breaks, which is by homologous recombination. And in homologous recombination, we make the break in the same way, but we have to provide a piece of what we call donor DNA that has a structure in which it has sequence homology to the region around the break and then sequence changes. And the recombination machinery senses or utilizes this undamaged donor DNA, and through a copy and paste mechanism, will make a copy of the donor DNA, paste it over the break, and we can thereby create either a single change in the nucleotides of the genome, or we can insert a large cassettes of genes. And by making the break and by optimizing the nucleases and the donor DNA, we go from not 1 in a million, but I'll show you up to almost 50 to 80% gene correction. So the idea then is can we use genome editing to revert the pathologic GTG to the non-pathologic GAG? And so I'm going to describe how we've used this as a way of developing a therapy for sickle cell disease. But I'll also say that the homologous recombination process allows us to do more than just correct single nucleotide mutations. It allows us to insert genes back into themselves as a way of one strategy fixing a multitude of mutations, because most genetic diseases, unlike sickle cell disease, are caused by multiple mutations in the gene, not just a single mutation. One can use this process to insert a gene cassette, a promoter and its own gene, into a specific location in the genome, a so-called safe harbor, which might have some utility. And then I will talk about the idea of taking a gene and putting it into another gene and starting to rewire how the cell works. And I'll talk about how we've built on the work from Michelle Sadelain's group in terms of knocking a CAR into the track locus. The system we use is an ex vivo system. We do this all outside of the body. And the first thing we do is we purify our stem cell or cell type of interest, and we have to put these cells into cycle. And the reason we have to put these cells into cycle is that the recombination machinery is most active, or perhaps only active, in S and G2 of the cell cycle. Once we put the cells into cycle, we then deliver our engineered nuclease, and we use the CRISPR-Cas9 system as a ribonucleoprotein complex, whereby we take the Cas9 protein, which is purified, complex it to a guide RNA, which tells the Cas9 nuclease where to go, and we deliver that RNA protein complex into the cells via electroporation. And that is a really efficient way of making breaks and indels. We immediately chase the electroporation with a viral transduction with an AAV vector. Now, AAVs are non-pathogenic viruses that have evolved ways of delivering their single-stranded DNA cargo to the nucleus with minimal detection by all the cytoplasmic sensors that our cells have looking for foreign nucleic acids and viral infections. It is a non-integrating virus. So as the cells divide, the virus disappears. So this is a way for us to get that donor DNA into the cells for a hit and run type approach. And this system is highly effective, and we've shown that it works in a number of different cell types. And I'll show you today the efficacy in hematopoietic stem and progenitor cells and T cells. So I'm going to describe in sickle cell disease how we're going to rescue an underlying missing function. And at the end, again, I'll tell you how we can add a new function using this. And so we first demonstrated the efficacy of correcting the sickle cell mutation in a publication in 2016 led by Danny Dever, Rasmus Back, and Joe Camarena. And what our donor vector had several features to it. Of course, the most important of which is that we revert the T back to an A. The second most important of which is we changed the nucleotides of the guide RNA binding site to prevent the RNP complex from recutting at the site after we've done the gene correction, because we and others have found that these nucleases would just come back and recut. And so these are silent synonymous nucleotide changes, so we don't change the protein sequence of the beta globin gene. And then finally, we have a short number of synonymous changes to facilitate recombination between the break site and the nucleotide change we want to make. And using CD34 cells from 6 different sickle cell disease patients, we went from 40% allele correction, so 40% of the genes in the population corrected, up to 60 to 75%. And I was really proud of myself that I can insert my favorite eye rolling emoji in here, because we don't really know how we got from 40 to 60%, except people just did it more and more. That practice makes better. And then I'll describe the importance of Annalisa later on. When we take those CD34 cells from sickle cell disease patients and differentiate them into red blood cells in culture and measure the proportion of the pathologic hemoglobin S versus the non-pathologic hemoglobin A, we find that, of course, without any manipulation, these cells make entirely 100% hemoglobin S in terms of the beta globin. In vitro differentiation creates some fetal hemoglobin, but we sort of ignore that because it's an artifact of the in vitro process. And when we take our gene-corrected population and differentiate into red blood cells, we're now creating 90% hemoglobin A and less than 10% of the pathologic fetal hemoglobin. So the next thing we need to ask, though, is we've taken these cells through a relatively complicated process. We've purified them, we've put them into cycle, we've shocked them with electricity, we've made a double-stranded break, we've delivered a virus to them, we've asked them to undergo homologous recombination. So have they lost their stem cell properties? And with that, I want to cite George Box, who on his Wikipedia page is described as one of the greatest statistical minds of the 20th century. I believe this was written after he died, so probably he didn't describe himself this way. But I cite Dr. Box because he has several quotes along the same lines, which is that essentially all models are wrong, but some are useful. Or a more convoluted way of saying is, remember that all models are wrong. The practical question is, how wrong do they have to be to not be useful? I like the first one better. The reason I say this is that the ability to analyze whether you have a true human hematopoietic stem cell can only be fully tested when we take a human cell and put it back into a human. But the FDA rightly won't let us do that unless we somehow have some evidence that that these stem cells still have their stem cell properties. So we have to use immunodeficient mice. It's a long buildup to say that when we take our genetically corrected human CD34 cells, we get different results in these engraftment experiments into NSG mice, depending on where the cells came from. So if we take CD34 cells from the peripheral blood of sickle cell disease patients in the spontaneous state, so they have not been mobilized out of the bone marrow, we get 60% gene correction. You saw this. And when we transplant these into mice, the gene corrected cells still remain very high, close to 60% cells. When we take mobilized peripheral blood CD34 cells from people who don't have sickle cell disease, we achieve the same frequencies of gene correction, and we can measure that, of course, through these other nucleotide changes because the A stayed the same as the A. And we put these into immunodeficient mice. We find that there is a drop in how many gene-corrected cells engraft, with some mice having very high levels of gene correction and some mice having very low levels of gene correction. Finally, if we take mobilized CD34 cells from sickle cell disease patients, and we got these cells from John Tisdale, again, the same level of correction before the cells went into the animal, but after the cells went into the animal, now more consistent levels of engraftment of the gene-corrected cells. We're pleased that even with this high variability in this mouse model, we mostly, particularly with the sickle cell disease CD34 cells, stay above what I call the Tisdale-Fitzhugh line of 20%. 15%, I use 15% here at the allele level because 15% of the alleles corrected would lead to somewhere between 20 and 30% of the cells being corrected. And one of the reasons that this mouse model, I think, is the best available model, but still relatively poor, is that we and the Naldini lab have shown through barcode experiments that the engraftment of human cells into these mice is highly oligoclonal, with only 1 to 5 clones really providing the bulk of the human cell engraftment. So there's a huge stochastic variation between mouse to mouse, even if you're using the same population of CD34 cells. Okay, so we were pleased with this. And then, one of the great things at places like WashU and Stanford is you have colleagues who have expertise and that complement your own. And so my lab is next to Hiro Nakauchi's lab, and in Hiro's lab was a super talented postdoc, Adam Wilkinson, who now runs his own academic lab at the University of Oxford. And he said, well, we have the ability to grow mouse HSCs. Let's test how well your system works in mouse HSCs. And we said, well, we don't want to design whole new targeting reagents. He said, well, we'll design some ROSA 26 reagents. And so he showed that when he knocked a GFP cassette into the ROSA 26 locus into mouse HSCs, the overall knock-in frequency was about 25%, so not quite as high as what we were getting in humans. But when he transplanted these into primary recipients and then secondary mice to really test functionally whether these were long-term HSCs, you can see that the GFP positivity was essentially identical to the original drug product of 25%. And the GFP positive cells were the same across red blood cells, platelets, myeloid cells, B cells, and T cells. So this was quite reassuring that the process in mouse HSCs weren't damaging their stem cell properties. And so we then took advantage of the Towns sickle cell mouse model, in which Tim Towns and his colleagues have replaced the mouse globin genes with the human globin genes. So the human alpha globin genes and the human beta globin genes, and specifically the human beta globin sickle cell allele. And so again, Adam and Danny and Job could show that they could get 20% gene correction in the mouse, Towns mouse HSCs. So not as high as what we were getting in human cells, but we hadn't spent nearly as much time optimizing the process in mouse cells. And then he transplanted these cells into basically syngeneic, in a syngeneic setting, and found that if the allele correction was 20% in vitro before transplantation, here we go, right, shown here, it dropped only a little bit across myeloid B and T cells. And excitingly, and there was, again, some variability with some mice having low levels of donor engraftment, generating only moderate levels of hemoglobin A between 20 and 40%. But a few mice with engraftment levels of 20% gene correction leading to 60, 80, or even 90% hemoglobin A. So again, showing in principle that we might be able to treat or even cure a human using this approach. This demonstrates that red blood cells that are not sickling have a prolonged lifespan, and thereby, even though there is lower levels of stem cells correction, there's much higher levels at the red blood cell level. So we took our process in the lab and worked with our GMP facility to scale it up to clinical scales. I'm showing here our moderate scale numbers where we're modifying between 20 and 150 million cells at a run. And the overall levels of correction are lower than what we were getting in the lab, which isn't entirely surprising, but still above that 20% number that I mentioned before. We did a lot of safety studies, including molecular safety studies looking at potential off-target sites, and found that there was only a single off-target site that had measurable insertions and deletions. This is a site that is 95,000 base pairs from the nearest gene and of no known functional significance. We collaborated with Richard Frock to look for any evidence of chromosomal translocations, and we saw that there was some evidence of chromosomal translocations between the on-target site and the off-target site using a wild-type Cas9. But when we used a high-fidelity form of Cas9, these translocations decreased by about 30-fold. And we complemented these molecular and genetic studies with a in vivo tox study in mice, and we saw no evidence that the cellular product caused abnormal hematopoiesis or cancer. And we recently reported this in Science Translational Medicine. I highlight Nihar Bhatia, who was the leader in the GMP facility, who just recently passed away of ovarian cancer, and just wanted to really highlight her contributions, important contributions to these studies. That led us then to submit an IND in September of 2020, proposing to use gene correction of a patient's own hematopoietic stem cells using the process I described, and transplanting them back into patients with severe sickle cell disease using myeloablative busulfan conditioning. And after the usual back and forth with the FDA, our IND got a may proceed in October of 2020. And then after lots of more administrative hurdles, which I won't go into, the trial was open both at Stanford and one day later here at WashU to begin treating patients with sickle cell disease. This is a cartoon that was drawn by professional artists trying to depict the journey that a patient will take as they move through this process, from identifying patients and harvesting their stem cells to manufacturing the cells in a GMP facility, to then infusing the cells through going through quality control on making sure the product has the characteristics that we want, to infusing the product through an autologous bone marrow transplant, and then following the patients for many years to follow. think that the artist did an amazing job capturing some of the really subtle and beautiful characteristics of what I hope will be a successful trial. Okay, so in the last few minutes, I want to cover two more topics. The first is to go into how we can use this not only to correct an underlying genetic mutation, but add a new therapeutic function. And then the last few minutes, I'll just touch a bit on my involvement with some of the ethical discussions around the use of genome editing in humans. And so this is the idea that I mentioned earlier about putting one gene into another gene. It builds on the work, as I said, from Justin and Jorge and Michelle, who demonstrated that they could take a CAR targeting CD19 and knock it into the TCR receptor alpha locus, the TRAC locus, and generate a product that they thought had better biologic properties. It turns out that it's probably more complicated than this because not everyone's been able to replicate it, but nonetheless, the idea is that the CAR is now controlled by the TRAC locus rather than by an exogenous promoter. So in collaboration with Alice Bertagna, who's one of the world's leaders in alpha beta depleted haploidentical transplant, and a very talented postdoc in my lab, Volker Wiepking, who has moved on to Genentech, we decided to apply this idea to alpha beta cells as part of an alpha beta depleted haploidentical transplant. Or we haven't done this clinically yet, so let me not get too far ahead of myself. So that was the concept. So in an alpha beta depleted haploidentical transplant, the alpha beta T cells are depleted. They're bound to a Miltenyi column and normally discarded. And so these types of transplants are actually highly effective in treating leukemia with outcomes that are similar to matched unrelated donor transplants and mismatched unrelated donor transplant. However, the overall survival is only 60%. So there is certainly room for improvement here. And so the concept is can we give, and one of the major reasons that it's only 60% is there's some fraction who relapse after transplant. And so our concept here is to give post-transplant CAR T cells to consolidate the anti-leukemic effect of the alpha beta haploidentical transplant by knocking out the TCR, so we do not cause graft versus host disease, and knocking in a CAR. And of course, we can achieve both of those by using the Sadelain approach by knocking the CAR into the TRAC locus. So again, the idea is we're going to create a cell that does not cause GVHD, but is also now not going to be rejected by the host immune system because we're using the alpha beta cells that are coming from the donor themselves. So they're going to be immunologically identical to the host immune system that's going to develop out of the alpha beta transplant. And so again, we'll knock the CAR into the TRAC locus. And what Volker was able to show, again, with practice, is that he could get knock-in efficiencies in T cells of 80%. And you can see the reproducibility across multiple alpha beta donors, so highly reproducible. And then with of course, an accompanying significant decrease in the amount of TCR on the surface. He could then run, these cells would proliferate reasonably well. They weren't quite as good as mock cells. And then he could take this population and rerun it over an alpha beta depletion column and create a product that now had only 0.02% TCR positive cells. So extremely low risk of GVHD. That now, before the alpha beta column, the TCR positive cells was somewhere around 3 to 5%. So maybe that's fine. And certainly Allogene has shown that a population of cells with 3 to 5% TCR positivity doesn't always cause GVHD. But we played the ivory tower idealistic academic and said we wanted to get it down even more. Interestingly, one of the evidences that these cells had activity is our discarded product from the alpha beta depletion actually also contains B cells. And we could show that once we manufactured the alpha beta cells to have the CAR, they depleted the B cells in the culture compared to the unmanipulated controls. So these were showing anti-CD19 activity. And then in a mouse model, when we injected NOM6 cells into the NSG mouse and treated those mice either with unmanipulated T cells, the mice quickly succumbed to the NOM6 CD19 positive tumor. When we used a lower dose of CAR T cells, 1 million cells per mouse, there was a delay in the progression. But when we used 5 million, a higher dose of genetically manipulated CAR T cells or genetically manipulated T cells with the CAR, there seemed to be complete cure of these mice up to 14 months with no evidence of xenogenic GVHD. So the key features is we engineering them from healthy T cells that are normally discarded. They're not going to be allogeneic with respect to the new immune system. They're engineered to not cause GVHD. They retain their anti-leukemic potency, and they have the potential to enhance disease-free survival. So now we're in the process of doing our scale up and process development to test the hypothesis whether administration of these cells post-transplant might improve the disease-free survival in patients with CD19 positive ALL following an alpha beta haploidentical transplant. So the two things I want to end with, and I think I'll leave enough time for some questions, is two ethical issues. The first is the ethics of equity and distribution. Obviously, academically, we can all be successes if we treat patients at places like Packard Children's Hospital, which I show here, or at the Children's Hospital here, and cure patients of leukemia and sickle cell disease. But as most of you know, most patients with sickle cell disease are in sub-Saharan Africa, where there's estimated to be somewhere between 250 and 300,000 births per year, and in India. So how do we bring these advanced therapies to parts of the world where it's not just by luck or happenstance that one happened to be have access to therapies at Stanford or St. Louis or other major medical centers? I will say, while this is a challenge, I also am optimistic that such one and done therapies in a way might be easier to bring to parts of the world than a chronic therapy that has to be given every day or every week. So we'll see. And of course, one can argue that, make the analogy that perhaps one-shot curative therapies is an iPhone and old school small molecule therapies are landlines, and that we can just skip over landlines if we can get to one and done therapies. I think all of us who do biomedical research have to be optimists, or otherwise we'd just go crazy. The last ethical question I just want to quickly discuss is the broader use of genome editing. And I was very fortunate to be part of a committee convened by the National Academies of Science, Medicine, and Engineering, who put out a report in 2018 about the science, ethics, and governance of human genome editing. And this has been followed up by further reports, both from the National Academies, from the WHO, and from the Royal Philosophical Society. And there was a lot discussed, but I think what really caught people's most people's attention was a consideration of whether genome editing should be used in a way in which the changes would be passed from one generation to the next, so-called germline or heritable editing, either to treat or prevent disease or for the possibility of enhancement, whatever that means. Well, I do know what that means. And our committee, these are questions that have been considered for decades by bioethicists, but the CRISPR-Cas9 system was beginning to have evidence of specificity to make people think that these sort of applications could actually be technically feasible. Now, the reality is even in 2021, they're still not technically feasible. But the committee made a conclusion that was slightly different, actually very different than what other ethicists had proposed, in which we said there would be, under very specific circumstances, a case to be made that heritable editing should be permitted. And And we proposed a number of very strict criteria. Now, in retrospect, I wish we had called it a functional moratorium to say none of these criteria had actually been met, so they were really in theory only. And the three criteria that I want to highlight is the first is that if a couple knows that they're at risk of having a child with a genetic disease, there is another way for them to have a child without that genetic disease that does not involve genome editing, and that is in vitro fertilization with pre-implantation genetic diagnosis. Now, in the US, a couple has to pay for that out of pocket. But in the UK, they have a list of diseases, which I believe is about 100 long, in which if you can demonstrate that you're on that list, the UK healthcare system will pay for you to have IVF with PGD to have your own children. And I think that that is really, I think that's a great system. The second criteria, which is actually, I think, unrecognized about how strict this is that you should only be able to edit to sequences that are known to be common and healthy in the human population. So what we did in sickle cell disease, where we make not only the change of the T back to the A, or the A back to the T, we also introduced some silent mutations. This criteria would say we couldn't do that, because those silent mutations are not in the human population. And therefore, we don't know that they're actually safe if they were passed along through future generations. And then the final point is that anyone who attempted to do this would have to have broad ongoing participation and input from the public. That doing this in secret or doing this would not be an acceptable way of proceeding. Which all leads me to conclude that what Zhangqi Ha did in China violated all of these criteria, despite his personal assessment that he was not violating the criteria, and was an egregious violation of the responsible conduct of research. John Cohn did an extensive analysis, as has many others, and I think has correctly identified that JK was not a rogue scientist. He actually talked to a lot of people and probably had sign off from more people than he has admitted, I think, for various different reasons. So with that, I want to thank you for the opportunity to present the Schechter Lectureship here at WashU and the opportunity to do this in person. I would get to work with an extremely talented group of people in my lab, clinician researchers in my Division of Stem Cell Transplantation and Regenerative Medicine, my colleagues at the GMP facility, funding sources from everywhere we can get money. And we really love collaborating with our academic colleagues at Stanford, academic colleagues from other universities, and we've been very successful in collaborating with biotech. And with that, again, thank you very much, and hopefully there's some questions.
Let me open it up for questions inside. Yeah, Dan.
Okay. Yeah, so the question is in the CD34 hematopoietic stem and progenitor population, that's a very heterogeneous population, undoubtedly. So we have taken that heterogeneous population and using standard cell surface markers, separated into MPPs, LMPPs, and phenotypic HSCs, and measured the frequency of gene correction in those different populations. And in the phenotypic HSCs, so 34 positive, 38 negative, 40, the whole list, the frequency of correction is about 50% compared to 70%. So it's still active. But I think what we're, all of us are a little bit worried about, though, is we're asking these cells to come out of quiescence, go through at least one round of the cell cycle, and will they have lost their stem cell properties? I was talking last night that we actually include the compound UM171 in our mix to try to preserve that stem cell property while we keep these cells in culture. Thank you, Dan. Yeah, sure. I don't know. Yeah, please. Yeah. So we didn't do those studies because they did a lot of those in Michelle's study. And clearly, so one thing I can say is when you knock in a gene into a specific locus, you get a pure population of cells. And when you use a lenti or retroviral and you have random integrations, that's for sure. What Michelle's group showed is that knocking into the track gave sort of a lower level of expression than when you use different exogenous promoters, even at the same locus. So clearly, it's a way of controlling the level of expression. His work demonstrated that with this CAR, there was lower levels of exhaustion than using other approaches. When we did it, it wasn't in our, I think it wasn't in our paper. We saw not such dramatic results. So I think that exhaustion knock-in approach is CAR dependent, and yeah. Great, thanks.
Yes, Johnny.
Yeah. Thank you, Shalini. So I'm going to repeat the question and rephrase it slightly and give a little background. For those of you not embedded in the field like the two of us are, recently there's been a report that a couple patients treated on a gene therapy trial, not using gene editing, with sickle cell disease, developed myelodysplasia following their therapy. And the question is where did that come from? And the answer is we don't fully know yet. The hypotheses are that sickle cell disease patients, because of their hyperactive marrow, are more prone to develop a pre-malignant genetic states. I don't think that's panning out quite so much in these patients, but it is maybe panning out in the population at large. The hypothesis is that when you do an autologous transplant, even using busulfan conditioning, you leave behind some stem cells that now have been exposed to busulfan, aren't fully eliminated, and that they can then reconstitute, and you develop the MDS not from your drug product, but from the remaining stem cells. I think that's a reasonable hypothesis, and why yet another reason why we want to move to non-chemotherapy-based conditioning. And then I think there will be some patients, and some of the new other lentiviral trials are showing that yes, lentiviral insertions can land in the wrong spot and be contributory to this, to the potential of developing MDS. I think in the long run, we were talking about this last night. I think we would love to move to younger stem cells. Younger stem cells because they're more potent, and younger stem cells because they haven't been asked to go through decades-long hyperproliferation in an inflammatory environment, and younger so that they might not have acquired those changes. So I think we need to show safety and efficacy in adults, but I think in the end, there's a lot of rationale for moving to younger patients. Yeah.
There are definitely some questions. Dr. Porteus, I want to thank you so much for being here with us today. First one we got here asks, what amount of off-target CRISPR-Cas9 activity did the FDA set as a safety threshold?
Yeah. So did they hear you, the question, or should I repeat it? Okay. So the FDA has not set a threshold of what is safe or not safe. And I think that's the correct analysis because we don't know if there's a threshold. What they have asked is that you use independent methods to evaluate the genotoxicity of your process and report on it and follow it. But they did not imply that if you have above this, it's not safe, and below that, it is safe. So this is a really ongoing field. What I will say is I believe the very first FDA-approved genome editing therapy came out of City of Hope, in which they were using zinc finger nucleases to inactivate the CCR5 gene as a potential treatment or potential way to develop HIV resistance. And their off-target profile was like, we wouldn't even tolerate that at all. Multiple sites with high, single digit or more frequencies of indels. And the FDA passed that because they didn't say that there was a threshold that they just said, we want to know it and we want to do a functional test and we want you to follow it. I'm sure that's going to change over time. Yep.
Wonderful, thank you. Dr. Lay echoes our thanks and says, would you like to contrast the beta globin gene correction approach you're using with the approach to modify fetal hemoglobin levels by inactivating BCL11A?
Yes. So first of all, I hope all the approaches work, right? The patients need choices and we have more than enough patients for everything to work to treat them. So what Dr. Lay is referring to is there are several, one major clinical trial from CRISPR Vertex, but others on the horizon in which you don't need to harness the homologous recombination process, but instead need to harness the non-homologous end joining mutation process by mutating an enhancer element in the BCL11A gene, or actually other programs are looking at directly mutating the BCL11A binding sites in the gamma globin locus to de-repress gamma globin. What we know is that when you de-repress gamma globin and create fetal hemoglobin, that's a countermeasure against the sickle hemoglobin. And patients with sickle cell disease who have higher levels of fetal hemoglobin have less severe disease. And so these approaches are trying to capture that. There are people in the world who have hereditary persistence of fetal hemoglobin and seem to be healthy. And so there is a rationale that creating high levels of fetal hemoglobin is a safe approach, but it is not normal. And we don't know all of the manifest. Somebody with high levels of fetal hemoglobin, because of their high affinity, probably, there may be some exercise intolerance issues and ability to really meet maximal physiologic capacity. It's way better in sickle cell disease. Let's not. Whereas what we're doing is saying we're not going to create an abnormal state of high levels of fetal hemoglobin like one has when you're just born, but instead we're going to create, hopefully, a profile of hemoglobin that looks like what normal adults have, or maybe sickle cell trait people have. We'll see. I think we'll see after 2 years and 5 years whether there's what are the risks and benefits of both approaches.
I have a couple of questions.
Yeah.
And then I think maybe one last at the back there. So one of the questions I had was, for the experiments using track knock-ins for 19 cars, one of the other, really the bottom line is whether they'll be cells will persist long enough to eliminate a re-challenge.
Mm-hmm.
So I'm wondering if you've actually looked at that using a track knock-in versus a traditional approach and re-challenge the animals with not. So one of the reasons maybe that knock-ins persist longer is that they don't expand as quickly and they don't undergo exhaustion, but there may be real benefits to that long term and that they're around for a much longer period of time. The other question I have to do with, has anybody actually been able to identify an acute and non-toxic way to put selection pressure in a genetically modified stem cell?
Yeah. So to answer your first question, we have not done the re-challenge in vivo experiment. We've done the re-challenge in vitro experiment, and they do respond to challenges in vitro, but not in vivo. I think the idea of putting selection pressure on the HSCs is really intriguing. We're starting to think about how one might do that. The other side, the other side of that is not to put too much pressure to create oligoclonal hematopoiesis too early in life. I think polyclonal hematopoiesis is a protective factor. So can we get the Goldilocks effects of just enough to enrich to where we want, but not so much that we create some oligoclonal hematopoietic system that is going to be prone to transform over time.
Last question.
That's a great question, and we could talk for days. So I'll give you a couple thoughts. It was really disappointing to me that Bluebird has withdrawn its gene therapy products from Europe because they did not feel they were going to get reimbursed at the level that they had modeled. We feel, my group feels that that's highly disappointing, that if you can't figure out how to, with a business model where you're still going to get $1 million for your product, you probably need a new business model. So that's not quite your question, but I think this is an active area of interest. In terms of how you get to Africa and India, I don't know all the answers, but here are some thoughts. Christian Medical Center in Vellore, India has demonstrated that they can do allogeneic transplants for $10,000 to $20,000, whereas they cost $200,000 to $1 million, $200,000 to $1 million here. So I think these really talented and motivated clinician researchers can show how you can do this in a cost-effective way. I think we need to put our processes in their hands to go through that. The final thing I'll just say is we need to take this all out of a GMP facility where I should show, you know, if some of you know, you're in these bunny suits and masks, and there's a lot of, we need to close it down, put it into a device where that could be parked in many different locations. I think closed system manufacturing is one way of reducing costs. I think that the cost of goods for our process can be brought down to a quite reasonable level. And I've had some discussions with the Gates Foundation that that can be done. So, yeah.
Matt, thank you very much.
Thank you,
for a
John.
Terrific talk and for coming and watching these questions.
Thank you. Is that right? Okay. Yeah. All right. Well, I just started some conversations with them.